everyone. So yesterday I gave a talk on tools that we use to make iPS cells and differentiate them. So today I'm going to talk about a technology that we have to be able to analyze those cells, your stem cells or any other cells for that matter. Sorry. It doesn't work?
Yes, it does.
Okay. So at Thermo Fisher we have reagents for a lot of reagents for protein analysis. We have a lot of reagents for genetic analysis. And so this technology, proximity ligation assay, is an interesting technology that actually combines the two. So the thing to remember about this technology is that you use two antibodies and you have a qPCR readout. And so this combination allows you to do interesting things that you can do with traditional protein detection approaches, but it's not as elegant. And so I'm going to start off with a slide about just what we want with proteomic tools. Well, the first thing that you do with proteomic tools is you measure protein abundances. But you have to remember that in lysates, protein concentrations can differ by more than 1,000-fold, and so you want this wide dynamic range. You also want to be able to measure protein variations. A number of proteins have different isoforms with subtle differences. And then another thing you want to, that's nice to look at, is to look at protein modifications and protein interactions. And so this is more challenging to do with traditional protein detection technologies. And then finally, it's nice to be able to measure proteins on a large scale, like multiplexing, so looking at entire pathways or entire cellular systems. And so PLA, or proximity ligation assays, is a perfect union of combining two worlds. And so what you have is you have the simplicity, sensitivity, and broad assay dynamic range of qPCR. I'll go more into that in a few slides. But then you have the specificity of affinity-based protein detection reagents. It's a scalable, flexible technology, and because of this, because of this qPCR readout with different fluorescent dyes, you can do multidimensional analysis with DNA, RNA, or protein. And so the basic workflow for this is fairly straightforward. There's a binding step where you bind two antibodies that have oligos attached to them, and I'll go into more of these details, I think, in the next slide. These ligate together, and then based on the amount of antigen that you have, of target that you have, you'll get a qPCR readout. And so you can read this kind of like RNA expression analysis and get quantification data based on that. And so, yeah, this is just showing that there's two antibodies, two oligos, and then when they're conjugated, you get the signal. And so there's two different platforms for being able to look at this. I mean, one of the advantages of this technology should be that it's easy to use, and so most of the time we like to steer people towards this homogeneous assay, because in the homogeneous assay you have a lysate sample, and then you just add your probes, you add your antibodies to the lysate, you let it bind, and you read it out. You don't have to do an ELISA where you bind your antigen to the plate, you wash, you add the secondary, and then do the readout. And so this is simpler. In some assays where you need that broad dynamic range, where your protein concentration may be changed by 1,000-fold or more, you want to use, that might be where you do a solid phase assay. And this is more of an ELISA type of assay where you have an antigen that's pulled down by beads, and then you do the same reagents. So it's a similar workflow, but again, with this type of assay, you get about 6 logs of dynamic range. And so here's the design scheme for the oligo readout. And so with proximity ligation assays, you think of it as a Western or an ELISA, but instead of having chemiluminescence probe or some other probe, your probe is this DNA oligo. And so in this assay, there are two antibodies. Each antibody has a different oligo. So you have an oligo A, a left oligo, which is about a 60-mer, and this contains a site for a forward primer and a probe. And so then you have a second antibody that has your reverse primer or reverse or right oligo. This is about a 40-mer, and this is where your reverse primer sits. And so in your conjugation, you need to have both of these probes bind to the target. And so once they bind to the target, these oligos come within close proximity, and you can add a splint to ligate them together. And so once they're ligated together, you only need one molecule or a few molecules to ligate. But then you have this template that's created that will only be amplified if this comes into if this is connected together. So you get amplification of your forward and reverse, and then unquenching of your probe. And so that's basically how it works. It's a very simple workflow. You just need to do dilution into a plate, add the probes, let it sit at room temp for 60 minutes, and then you just add a ligation and a PCR mix together. And so this serves, you just have a 37-degree step where you have well, actually no, it's lower than that. It's 16-degree step where you just ligate that splint on, and then you start your qPCR reaction. And so this is comparing the workflow between an ELISA and the TaqMan protein assay. So again, as I was saying, for the ELISA, you have to actually coat your plate, have the sample binding, wash, add your detection antibody, and then your substrate, amplify, and then stop and read the OD. So with this type of assay, you just need to the analogous part is just probe binding and then ligation and qPCR. And so there are two ways to measure this out. So because this is qPCR based, you read out the signal by CT. And so you can look at this by absolute CT. So that's what the graph on the left is showing here. And so you have you basically have your range, your readable range right here, where you look at absolute CT, and the more, just like qPCR, the more signal you have, the lower the CT is, the lower your threshold will be. So this means the more input your CT goes down, or you can choose to have a reference and have a delta CT where you'll get the opposite type of graph. And so this is just this is an example of showing specificity for this type of assay. This is it's an example using OPG, osteopontegrin, and rank L binding. And so this is just an assay with recombinant proteins, showing how the signal that you get when you have different probe pairs and protein inputs. And so this is how the assay is supposed to work. What I'm showing here is a probe pair of OPG, right and left. And then I have there's an OPG input here, recombinant protein. And you can see that as you add more protein right here, your signal goes down, which you would expect. So we're just looking at CT right here. So the same thing happens if you have, if your probe pair is again only to OPG, but then you have a dual complex of OPG and rank L. Again, you're only looking at OPG, so your signal here is the same, or roughly the same as this. The same thing happens with rank L, if you look at a pure rank L binding. But then what's interesting here is that this is a little bit hard to do with traditional protein detection technologies. You can look at the dimer with this assay, and so you have a protein pair of OPG and rank L, as shown in this cartoon here, and then you add the probe pairs of one OPG probe pair and another rank L pair. And so when these come together, these can only come together if there's binding. And so you can see right here that this is an assay that can show the dimer, and so here, if you have if you only have OPG and you have an OPG and rank L pair, antibody pair, you will not get any signal right here. So it's showing that this assay is specific for what you're looking for. Okay, so this assay can be very sensitive because of this qPCR amplification. And so this is just showing IL-6 levels in human serum. And so this is showing a standard curve of different IL-6 amounts right here. And this is a curve showing what happens, or the IL-6 levels that are measured in different percentages of serum. And so your assay dynamic range here goes from 0.128 picograms per mil to 2,000. So you've got this broad dynamic range, but then your limit of detection is extremely low, a lot lower than you would get with traditional ELISAs. And so I'm just going to go through a few examples. One example, since this is a stem cell meeting, one example that you can look at is to look at the difference between embryonic stem cells and differentiated cells, neural cells, mesenchymal cells, or hematopoietic cells. And so yesterday I talked about markers of pluripotency, including NANOG, OCT3 or 4, SOX2 or LIN28. And so these are cells that were obtained from Solardis in Sweden, and so that's a company that makes embryonic stem cells. And so we got Entera-2 lysate, which is our embryonic carcinoma control, and then a Raji lysate, which is more of a lymphocyte mature cell reference. And so what we're looking at is the comparison of the stem cell markers, the 4 stem cell markers, in the Entera-2 cells versus the Raji cells. And so you can see that as you increase your cell input, you start to see expression of all 4 of these markers with the Entera-2 cells, but they're basically flat with the Raji cells. And so this is just showing a comparison of expression between Western blots and the PLA assay. And so in this assay, you use the same antibodies for the Westerns. Those same antibodies are used to make probes for PLA. And so this is just showing the detection limit that you get by Western blot with different inputs of cells. And so when these same assays are applied to the PLA assay, so one other another name that we give for the PLA assay in-house is TACMAN protein expression assay. And so compared to about 100,000 cells that you need for a Western, you only need about 100 to 500 cells for the TACMAN protein assay or the PLA assay. Your limit of detection is about 30,000 to 10,000 for the Western, and you can get down to about 10 to 35 cells for the PLA assay. So it's much more sensitive than traditional methods. And so this is just showing another example of expression comparison between pluripotent cells and differentiated cells. And again, these cells were obtained from Solardis, these SA002 cells, pluripotent cells, compared to these mesenchymal cells, MP002. And so when you look at expression of stem cell markers, this is just a comparison of stem cell and differentiated markers in the mesenchymal cells compared to the embryonic cells. You can see that the mesenchymal cells have a significant downregulation of these stem cell markers, and this basically looks like an mRNA qPCR expression plot, but this is protein. These are protein levels. And then when you look at upregulation of the mesenchymal markers, that a lot of them were talked about in the previous talk, you can see an upregulation in this with this set. Okay, and so this is just showing direct differentiation of Entera-2 cells with retinoic acid from into embryonic carcinoma cells to these neuroprogenitors. And so, so with this is just showing that you can multiplex. So you can take these lysates and you can use different kits to harvest protein, mRNA, or microRNAs. And so when you do that, you'll see a loss of when you see this loss of pluripotency, you see an increase in miR-145 expression, and then a decrease in OCT4 expression. And so when you follow the expression over time, over 14 days, you can see that this miR-145 starts to go up as predicted, but then the M4 mRNA goes down, and then likewise, when you follow the OCT4 protein by this TACMAC expression assay, it also correlates with this decrease in OCT4 expression. And so this is an example of looking at being able to look at protein post-translational modifications. And so in this case, we're looking at P53 phosphorylation. And so this is a little bit, these types of assays are a little bit trickier to do with Western blots or ELISAs. So in this case, with the ELISA, you're pulling their antigen now with total P53 protein antibody, and then you're detecting with phosphospecific antibody. And so you take these same antibodies and you make probes out of them, and then you can just add them. Oops, sorry. Instead of doing the ELISA, you can just add them straight to your lysate and read it out. And so this is showing what happens when P53 is phosphorylated by adding etoposide. And so in etoposide versus non-treated cells, you start to see this increase in ELISA of phosphorylation. And when we do the TACMAN protein assay, you basically see the same thing. The difference here is that your limit of detection is about 1,600 cells with the ELISA, and using the same primer pair, your limit of detection is basically reduced by an order of magnitude. And so I only have a couple minutes, or probably a minute left. So this is just an example of the same thing, looking at post-translational modifications of AKT. Here's another example of that. But in summary, this technology, this PLA-TPA technology, enables a broad range of protein detection applications. And I went over a lot of these examples, including looking at single cells, small numbers of cells, protein modifications, protein-protein interactions, and then serum plasma markers. It's an extremely simple workflow. You just add your probes in, you let it bind, and then you add it to the qPCR machine, and you read it out. It's a simple, flexible platform. It's scalable. You can do multiplexing if you have different reporters. This is just showing some applications that you can use with this type of technology. And just a little quote here. So just acknowledge the people who really pioneered this work. First and foremost, Mark Shannon, who really has been driving this technology and really been continuing to see it through. Xiaomin Chen developed a lot of the methodology, and the rest of the people really helped support these development efforts. Thank you very much. I'm happy to take any questions.
Time for one question.
What is the size, the distance limitation between the two epitopes for having your technique working?
The distance?
Yes. There is some limitation in the distance between your epitopes.
Yeah, there is, and that's actually one way that you can map it out. I want to say 10 nanometers, but I'm not sure. No, it's got to be less than that. I don't know. I'm not sure if it's been characterized, honestly, the exact distance, but they have to be. It has to be in close proximity in terms of being on the same molecule, being within maybe 200 amino acids apart. Because even if you have random floating fragments of different probes, those will not, I mean, if you think about that, just by random chance, they'll come close together, but that's not close enough for them to generate a signal.
So